Hello and welcome to the Solving Scientific Problems Using Video Games Disrupted Bytes with Dr. Seth Cooper. Seth is an assistant professor in the College of Computer and Information Science at Northeastern University, Boston, and a member of the Playable Innovative Technologies Lab with an interest in using video games to solve difficult real-world problems. He is the chief architect and lead designer of Foldit, a video game that has allowed hundreds of thousands of players to contribute to biochemical scientific research. We are pleased that Seth is today joined by our very own Sylvester, who is in Boston for his visiting fellowship. Seth will talk about games and citizen science, specifically the challenges of mapping real-world problems onto games and ways to address these challenges. As a primary example, he will discuss Foldit, an online game about biochemistry whose players have contributed to several scientific discoveries through gameplay. There will be time for questions at the end, but in the meantime, we will cross over to Seth in Boston.
Hi. So I'm just going to start off with some PowerPoint slides here. Let's see. Okay, can everybody see that?
Yeah.
Yeah? Okay, great. Then I'm going to get started. Thank you for the introduction, and thanks for inviting me to speak to your group. And I'm going to be discussing some of my work on using video games for solving real-world problems. I've taken this approach because games are very good at engaging players and training players in solving problems, but they're typically in problems that are made up for a game, just for entertainment purposes. So I'm interested in what we can do, what we can accomplish if we can take all the effort that players put towards solving problems in games towards solving real-world problems and real-world challenges. And I think that if we do that, we can use games as a means of combining the power of the human mind, of creativity and problem solving that people have with the raw computational power that we have available, and to be able to solve problems that neither humans nor computers would be able to solve alone, and use games as a means to sort of foster the long-term engagement that's necessary to gain skill in solving a problem in a particular domain, and also give players potentially new opportunities to and new pathways to work on things that are meaningful to them, ways to get involved in particular challenges that they may find interesting or relevant. But in order to do that, we first have to make a game that solves a challenging problem, a challenging real-world problem or computational problem. And so how do we do that? Well, typically the approach that I take is to build a game or a system that engages and trains players to work together with the computers, and then just starting from that, basically improve the game and the system over time, so that as time goes by, as we continue to work on the game, the players themselves become better problem solvers, and the game itself becomes a better problem solving tool. And so in that case, in that way, both sort of these two elements of the system are getting better and better over time and improving, and hopefully working towards being able to solve a real challenging real world problem and help out that way. So I'm going to talk a little bit about two scientific discovery games that I've been working on. The first one is called Foldit. Foldit is a multiplayer online game where the players are actually competing and collaborating to predict, to fold, and to design protein structures. Foldit was actually released in 2008, and yeah, since then it's had over 400,000 players. And Nanocrafter is a game that allows players to build basically nano machines and nano devices at kind of a molecular scale by assembling pieces of DNA and actually building sort of self-assembling systems of DNA that can create these sort of nano devices that could be tested in the lab. And so this game, Nanocrafter, was actually released in 2014 and has had over 10,000 players since then, since we released it. So these two games are sort of going to be the motivating examples behind some of what I'm talking about. So in terms of proteins, actually, why were we interested in building a game around proteins? So proteins are actually these molecules that are responsible for a lot of kinds of processes and mechanisms that happen in living organisms. And every protein is actually coded for by a sequence of smaller molecules known as amino acids. And so, relatively speaking, it's easy to determine the sequence of amino acids for a protein, but it's actually very difficult, even knowing the sequence of amino acids, what the three-dimensional structure of that protein is. And knowing the three-dimensional structure of the protein is actually very important for understanding the function of the protein and how it does what it does in the cell. And so actually, because of this difference in sort of the difficulty of determining sequences and structures, the difference, sort of the gap between the known protein sequences and the known protein structures is actually getting wider and wider every year. So there's this existing challenge, this large challenge of basically being able to determine what structure a particular protein will take given its sequence. And so there's actually been a number of sort of purely computational approaches to solving this problem, some of which you may have heard of. These two are distributed computing projects, Rosetta at home and Folding at home. These are screensavers. So when you are not using your computer or you're not using your PlayStation 3, they will actually sort of donate that spare computing time to helping to kind of compute to try to predict and simulate how proteins fold. At one point, I'm not sure if this is still true, Folding at home was considered the most powerful distributed computing cluster in the world, and all of that spare computing power was being put towards trying to compute how proteins fold, but it's still a very difficult and challenging problem to try to solve, even with all of this massive computing power. In terms of DNA synthetic biology, this is actually, and this is the domain that sort of inspired Nanocrafter. This is a little bit of a newer field. And so in this case, we're actually interested in more kind of trying to explore what kinds of devices and what kinds of structures are actually possible to be built using DNA as self-assembling DNA structures as a basis, as a building block, as a way to kind of build these structures. And so synthetic biologists have been working on this, and they've been coming up with a lot of interesting kinds of devices that can be built. And so in this case, we're interested in players' sort of creativity to see what kinds of things players will come up with, and if they can actually come up with new kinds of devices and new structures and new nanomachines that haven't yet been discovered. And so whereas Foldit has more of a sense of actually solving problems, solving specific instances of problems, Nanocrafter is more of a sandbox kind of feel to it to see what people can build and what kinds of things they'll come up with. Is the video working okay?
Pardon?
Is the video working okay?
No, it said that your slides are...
Oh, is it actually stuck on the... Like,
not showing the slides. It's showing the...
It's not showing the slides? Oh, okay. That's a problem. Is it just showing the...
Seth, it's just showing the whole screen. We can see all of your notes and everything. You just need to make it full screen.
Huh.
That's it. Click that.
Is it working now?
No.
Do you see it now?
No, we just see your window, your whole PowerPoint window.
I just
use this.
No, I mean, it should be possible to display the whole thing. Just click play, I think.
This one?
Yep. Something.
Slideshow? Okay, this is what I had done before.
Is it working, Alex?
No, you need to play the slideshow.
Yes, we have.
Yeah.
No, we're seeing what you can see. We can see what you can see.
it.
That's the problem.
I actually just see.
Yeah.
I actually just see the slideshow in full screen. Like I don't see any of the.
Yeah.
Maybe you're sharing the wrong window. See if you've got another option for showing a different window.
Okay, let's see. Yeah? No?
No, we just see your slide as a small window nail in the middle.
Slide.
Can you press from current slide?
Slideshow from current slide. Oh, what if this is broadcast show?
Try that one.
Oh. Huh? No,
No,
that's
it's not.
probably.
No, it seems to just be stuck like that. If you want, we'll leave it as it is, and then perhaps we can overlay the full screen slides afterwards.
Okay.
So you can go back to what you were showing. Yeah. Unfortunately, they're just not very big here.
Did that work? No, that's not any. That's not any better. Okay.
You just get the slides and you will find it with it.
Can I hide this? Yeah. Okay.
Yeah. That's fine. That'll do.
Okay. That's probably about as
Technology and
full screen as we can get them then.
No problem. I mean, we can edit a new recording with the full slides on afterwards. That's fine.
Okay. That's good. If we can go back and do that, that'll be great.
Yeah. No, we'll sort that out. That's fine. Okay, you carry on.
Okay. Cool. I'll continue. Right. So we have these problems in proteomics and DNA nanotechnology that we're interested in contributing to. And so they're difficult problems, but in the case of proteins, it's all about three-dimensional shapes. And in the case of DNA nanotechnology, it's largely about kind of understanding these systems and the pieces that how they work together. And so, players and people have a good understanding of like spatial reasoning and thinking about how systems work. And so we thought that these kinds of problems actually might make very good puzzles, and that we might be able to allow people to get involved and contribute things like their spatial reasoning and their understanding of how systems might work to, in the case of protein folding, augment the purely computational approach that some of the screensaver and volunteer computing approaches are taking. In the case of DNA nanotechnologies, to contribute some of their creativity to try to help contribute to solving these problems. So when we were thinking about how we might take these real world problems and turn them into puzzles for a game, we considered 4 sort of main areas of how to do this: the visualizations, the interactions with the problem, the goal that we wanted to give to players, and how we might train players to be able to contribute to these problems and understand the domains. So for the case of visualizations, a lot of what we wanted to do, or one of the main things that we wanted to do, was be able to actually abstract away a lot of the complexity. So you can actually see here, this visualization here is kind of a relatively common scientific view of a protein, and it's showing all of the atoms in the protein. And so it's actually fairly complicated. It might be difficult to see what's going on. So in Foldit, we actually abstract away a lot of the details of the protein. And so a lot of the atoms are kind of hidden, and they'll only show up when you mouse over regions, or when areas of the protein are actually causing trouble. And so in that way, we can actually hide a lot of kind of like the unimportant details of the protein, and hopefully show some of the overarching structure and make it easier for players to see and think about. Additionally, we added to the protein a number of different sort of external visualizations that float around the protein and show the players where there are areas that they might want to change, or areas where they might want to actually, things are good and they might actually want to keep things the way they are. And so these include things like clashes that show players areas of the protein that are too close together, and they might actually want to pull them apart. There are things like these bubble voids that show up in the interior of the protein when there's too much space, and players might actually want to try to compress the protein around those areas. There are exposed hydrophobic parts of the protein that are areas on the protein that would prefer to be kind of buried on the interior of the protein, but they might be exposed on the exterior. And so we kind of highlight those for players as well. And then there are also good things like hydrogen bonds that help to hold the protein together. And these are color-coded as well. You may notice kind of by their severity to some extent. Clashes and voids, you generally don't want to have any of those. Some exposed hydrophobics are okay, so those are yellow. And then the hydrogen bonds are generally good, so these actually show up as blue. So in the case of Nanocrafter, we also try to abstract away a lot of the complexity of the DNA that's being used to actually build the system. So here is what you might consider a full three-dimensional view of what a model of DNA might look like. And so in fact, scientists who use more abstract representations, including ones like these, where you just show two-dimensionally the sequences of base pairs and the complementarity. And then you can even get more abstract and abstract away actually the specific base pairs and just look at things as domains, sort of unique sequences of base pairs or their complements. And you actually just sort of show the directionality, the complementarity, and the identity of these domains and sequences of base pairs. And so we've chosen for Nanocrafter a visualization that's very similar to that and actually abstracts away here. You can see here a lot of the details of the underlying DNA system. It's only two-dimensional here. And again, it's just sort of showing these individual domains or short sequences of DNA, showing their directionality and their complementarity, and using colors rather than numbers or letters or anything like that to actually show the identity of these short sequences of DNA. And so this is what it looks like in the game. In terms of interactions, these are, in the case of Foldit, they're all based on direct interaction and direct manipulation of the protein. And so we actually did some early play testing where we had much more indirect kinds of methods of controlling the protein, where you would have sort of like menus with sliders and things like that, where you could control different properties of how the protein was folding up. And in some of this early playtesting, we found that it was much more intuitive and much more fun and enjoyable to actually be able to just directly manipulate the protein structure. And so we worked a lot on making tools for the game that allowed players to kind of feel like they're directly manipulating and kind of just touching the protein and able to move it directly by clicking on it and dragging and interacting. And even some of the optimizations that exist in the game are launched, again, by clicking on the protein itself, and then menus and things like that will come up from the protein itself to allow players to have this feeling of directly interacting with the protein and getting feedback that way. So in addition to having sort of intuitive and enjoyable interactions with the protein, because we're trying to actually solve a real world problem, we want the interactions to sort of be realistic and plausible and actually result in scientifically useful structures coming out of the player's interactions. And so because of that, essentially all of the interactions in the game are built around this biochemistry library that we built the game on top of called Rosetta. And whenever players are manipulating the protein in the game, essentially what happens is their inputs kind of get packaged up and turned into optimizations that get sent to this Rosetta software package to decide how the degrees of freedom of the protein should actually be updated to reflect both, to try to match as well as it can, both kind of the player's intention about where they're trying to move the protein structure, but also kind kind of the rules about how protein structures can move and bend. So you can't just make completely implausible protein structures. And so this Rosetta software package that's actually running underneath Foldit is the same software package that's running in the Rosetta at Home screensaver when you run, if you're part of the Rosetta at Home project and you're running the screensaver, it's actually running the same biochemistry modeling software underneath. And so actually in this way, we're able to combine, again, humans and computers, allowing players to do this high-level decision-making about roughly where the pieces should go. But then it's actually the computer and the Rosetta software package underneath that's able to determine exactly where every single individual atom goes and sort of refine the positions. And the players don't have to worry necessarily about getting the position of every single atom correct and plausible, because the computational engine running underneath takes care of that. Similarly, for Nanocrafter, we went with this more direct interaction and manipulation kind of interface where players can, here you can see basically a player building a little system out of DNA. You can actually create these little domains. And again, each one of these little pieces here on the screen represents a short single strand of DNA, and then when they're connected, that's two complementary pieces making a double strand of DNA. And players can create strands and connect them to make longer strands, and then also break them apart and delete them and so on. And so they can build structures and devices that way. And once they have something built, then they can actually simulate what would happen to this particular DNA if it were synthesized in the lab. And so you can see what's going to happen here is the player is going to click play, and then the DNA is going to start moving on its own. And what's going to happen here is a strand displacement reaction where you can see essentially these two complementary pieces of DNA are going to come together and bind, and that's going to cause these two red pieces to then bind, and this long piece on the top is going to come off, and then it's going to come over and bind to this piece. And so you'll actually see two what are called strand displacement reactions here in this example once it plays. So there it is. So there's the first one, and then the second one. And so this kind of strand displacement reaction is one of the key mechanics of the game that we built the game around to allow players to actually build these kinds of dynamic devices out of DNA that aren't just static, but that actually move, and they can sort of form these cascading reactions and do lots of interesting things, as we'll see in some of the examples later on. And so again, the interactions, this strand displacement kind of reaction that happens in Nanocrafter is a sort of a common reaction that's used in modeling DNA synthetic biology systems. And so we actually have a really powerful simulator of DNA dynamics that's running underneath the game. And we're sort of striving for, with the simulator that we've written, there's sort of this standard DNA simulator called a Visual DSD that exists. And with the simulator that we have running in the game, we're sort of striving for parity to some extent with that state-of-the-art Visual DSD simulator, so that the simulations of the DNA dynamics and strand displacement reactions that happen in the game are as accurate as they can possibly be, so that if someone builds a system that works in the game, we can hopefully be confident that, or as confident as that we can be, that it would actually work in the real world if it were synthesized in the lab, similarly to how it works in the game. So next I'm going to talk a little bit about the goals in both of the games. In Foldit, what we're trying to do with the score is actually give players a goal that directs them towards an unknown solution. So in the case of players folding or designing proteins, we don't actually know what the correct structure is. So we don't actually know what the score we would want players to achieve is. But so what we've done is we've actually we can get from the Rosetta software a measure of the energy of the current structure that the protein has. So it can essentially look at the current state of the protein and give back a number that says this is the energy of the protein, but we don't know what the best energy that that protein could have is, what the best fold could be. So we took the energy and we, instead of trying to get a low energy, which a better energy is a lower number, we want the players to try to get a higher score. So we took the energy, we negate the energy, and then we scale it up by a large number. So instead of trying to get a low energy, players are trying to get a high score. And so in this way, the score that the players are seeing as they interact with the protein is a good reflection of the quality or the energy of the fold that they currently are working on. And so again, because we don't actually know what the best score or the best energy we want the players to get is, instead of setting some particular target score for the players to achieve or something like that, we actually organize the whole game as a leaderboard competition where the players are essentially competing, and as they're working on their protein, as they're folding, they can see the score that they've achieved, and they can also see the score that all the other players who are working on that same protein structure have achieved. And so in that way, the players are sort of continually setting higher and higher goals for each other, and we don't necessarily have to set a particular limit on score to see what players will find. And in addition to competing, we also set things up so that the players are able to collaborate, and they can actually form groups and work together. And so when the players are in a group, essentially what they can do is take the protein structure that they're working on, and they can share it with their group members who can essentially pick up where they left off. And so multiple players can work on the same protein structure, improving on the work that each of the other players has done. And this is a way for the players to collaborate. And then there's also group leaderboards as well. So there's competition within the group. So there's sort of multiple forms of competition and collaboration within the game. In Nanocrafter, the goals are much more open-ended. And in this case, not only is the solution unknown, but we don't even really have like a nice objective function or way to automatically evaluate the structures that the players come up with. So actually, in the case of Nanocrafter, the goals that we give to players are actually very open-ended. In this form, they're text prompts where we run, when we give players a problem that we want them to solve, we actually just give them kind of a text prompt that they can then respond to by building some kind of structure. In this case, this is actually one of the simpler kind of drawing challenges that we present to players. In this case, the prompt is just build a pie. This is not necessarily a scientific challenge that we would pose to players, but more for them to have fun. And so we don't have a way to kind of automatically evaluate whether they responded to that prompt appropriately or in an interesting way. So the way that the players actually get points in Nanocrafter is through ratings by other players. So once a player has created what they think is a good solution to a prompt, they can upload that and share it. And then other players can go through a gallery or kind of a slideshow watching other players' solutions and then rating them whether they think they're practical or original. And if they get a point, if a solution gets a checkbox for both practical and original, then they get a bonus point for being creative. And so players want to build, or to get points, players are trying to build structures and devices out of DNA that respond to the prompt and that other players will actually vote on. And so there's a gallery that players can go to, again, to kind of see all the other solutions that players have uploaded, and they can view them, rate them, and so on. And so in this way, we're really able to have a kind of an open kind of sandbox feel where players are responding to very open-ended prompts, and they're sharing the things that they've made with other players who can see them and then rate them. And so in this way, we're sort of crowdsourcing through the game, not only the solution to the problem, but also the rating of the kinds of solutions that players have come up with. In addition to the open-ended kind of prompts, we have some in the tutorial levels for the game, which I'll actually get to in a second. We have sometimes we want to indicate particular pieces of DNA that we want players to interact with. And so if we have like a specific goal of something to do with a particular piece of DNA, we can actually mark those with a star to give players an extra goal. And I'll actually talk about that a little bit more, talking about the tutorials. So for training in Foldit, we have a sequence of introductory levels that introduce the different concepts for trying to introduce the game to anyone who doesn't necessarily have a background in biochemistry and kind of make the game very approachable. So there's a sequence of tutorial levels. They start out with very relatively simple proteins, small proteins with relatively simple things wrong with them that players can fix. And they have these text bubble hints that come up and they guide players through the steps necessary to kind of fix this, what might be wrong with the protein in this level, and help them be successful. And these puzzles, tutorial puzzles, also have, these do have fixed goal scores that the players are trying to reach. And so once they've fixed everything that's wrong with a particular protein in a tutorial level, they will have achieved the goal score and they can move on to the next level. And then again, the players are rewarded for success in the level with kind of fun animations. And the levels get more and more complicated as they go on, to the point where they're hopefully gotten to the point where they can compete in the puzzles with other players. Compete in the scientific puzzles with other players online and go into the leaderboards. Nanocrafter has a similar set of tutorial levels that, again, have relatively simple DNA systems that we want the players to build, and there's these hints that guide them through it, teach them how to rearrange the structure, how to break things apart, how to connect things, you know, how to run the simulator. And again, they get more and more complicated as things go on. And in these levels, these tutorial levels, again, so that we can know when the player has completed the level, we use these domains with the stars on them to indicate which domains the players are supposed to try to displace with the strand displacement reactor. And so you see in that case, it was unsuccessful. So the player needs to sort of rearrange that one strand, and then once all of the star domains are displaced, then the player is successful in the level. And again, these levels, again, with the DNA, they start out relatively simple, and they get more and more complicated, hopefully to the point where players have learned enough to begin working on and contributing to the scientific puzzles that we post. In addition to these tutorial levels in Nanocrafter, we actually have what we call drawing challenges, where they're not necessarily for scientific interest, but they're kind of in between tutorials and scientific challenges that we post, where we allow players to just kind of build different shapes with DNA and share those online and get ratings and votes and so on. Essentially, they have the whole same process as the scientific challenges, but they're more for allowing players to kind of get into the game a little bit easier, do some creative things, figure out how the process works. And so, for example, these are some responses that we got to the drawing challenge to draw something that looks similar at different levels of zoom. And so these are 4 responses that we got from players. So once we've started getting a game ready, or once we have an initial game, and we've got these goals, tutorials, visualizations, and interactions that we think will work, we don't necessarily expect or presume that we got everything right the first time. So we actually set up the whole game as a process of continual refinement, where we can get, again, data from the players, and we can try to continue to refine the game to make the game a better problem-solving tool, even while a community of players is learning about the game and how to play and becoming better problem solvers. So typically what this will look like is we'll take some open problem of interest that we're interested in seeing how the players are able to do. And we can turn that into a puzzle in the game and post that online to the the infrastructure that we developed for the games. And then those puzzles are sent to all the players who are playing the game all around the world. They can see those, and in the case of Foldit, try to fold a protein and see their place on the leaderboard. In the case of Nanocrafter, they can see the prompt and they can submit their solutions, and they can rate other players' solutions. And typically, we leave these puzzles, these scientific puzzles, up for about a week, maybe 2 weeks, depending. And during that time, the solutions that the players are generating are then reported back to the infrastructure, the database, so that they're stored and logged. And then we can get, from that, we can get aggregate solutions back, and we can do some sort of analysis depending on the particular type of puzzle that we're looking at or the particular type of problem, and see how the players did, what kinds of solutions they come up with, if they solved the problem. If they didn't solve the problem, then what kinds of things might we be able to change to try to help the players do better the next time through. And so that information then is fed back into the next round of puzzles. We might post the same problem with a different puzzle setup that might be used to help hopefully improve the kinds of solutions that the players come up with, maybe more information, maybe more constraints on the kinds of things they can do, maybe fewer constraints, and so on. And then we're also posting, for example, updates to the game if we need to add some entirely new feature or fix some bugs or something like that, maybe add some new levels. If we want to try something totally different, the games can be updated to add new things. And in addition to the scientific data, we can also get back gameplay data, for example, how far players make it through the tutorial levels. And so we can see when new players come in, when do they quit, for example, and can we do anything to improve that? So one example of something that we had done using that data for the tutorial levels was we looked at how the help system in the game affected how far players got through the tutorial levels to see what we could do to improve that and hopefully help players stick around more and possibly contribute more to the scientific problems in the game. And so in this case, we actually ran a test where we deployed 3 different help systems to the players. One of them was no help at all. One of them was what we called context-insensitive help, where it gave the players all the information that they needed up front in kind of a manual interface that they could page through and then close and then play the game. And then we also gave what we called context-sensitive help, where it was that system sort of that was shown in the video, where it was context-sensitive help bubbles that would sort of try to figure out what the player was doing right at that point in time and give them a targeted hint or feedback about what they should try to do next to complete the level. And so when we looked at how far on average players got in each of these levels, what we found was that with no help, on average players were able to make it through about 4 levels. With the context-insensitive help, they were able to actually get a little bit farther, but not much farther, only about 1 level further on average. Given all of this help information, but it was context insensitive. And then what we found, though, was that with the context sensitive help, they were able to get on average through 7 levels, which is almost twice as many as the not giving them any help at all. And of the different help systems that we tried, that was the best one. So that's actually why that's the help system in the game right now. But it's interesting to see how the context insensitive help here actually didn't help actually all that much in terms of seeing how far players got through the levels. We've also done a demographic survey to see who's playing the game, right? So we wanted the game to be accessible to people outside biochemistry who don't necessarily have a background in biochemistry, and so to see how just sort of get an idea of who's playing, we did a demographic survey a while ago, and these were some of the results for a few different question responses that we got. So you can see there are a large number of students playing, a number of people in the computer field, some people, you know, in art or business or legal. So it's interesting to see the backgrounds of a lot of the players. Another interesting thing that we noticed was that there's really a large international community playing the game. The largest, you know, the USA there is maybe, you know, 2/5 of the pie, but there's actually a large number of players who are coming from around the world. And in fact, because of that, we've sort of added support for translation into the game, and it's been translated into a variety of languages, including, I think, Russian and Polish and so on. We were also curious, or particularly about the player's background in biochemistry, and when we asked them about this, what we found was that about 3/4 of the players didn't have any background in biochemistry beyond maybe an undergraduate course. And we found that this was generally true of the players as well as kind of the top players, the players who are doing well. So we didn't necessarily see the case that, you know, the top players were all biochemists, but actually there was a lot of players who were doing very well in the game didn't have a large formal background in biochemistry. Okay, so that's kind of some background on the game. I'm going to talk a little bit about some of the results that the players have produced. I'm not sure I have a whole lot of time, so I'll try and get through this relatively quickly. Well, I'm going to talk about protein structure prediction, some results that the players have produced, algorithm development, and then protein design. So again, protein structure prediction is sort of what we started Foldit on, and this was given a sequence of amino acids, the sequence for the protein, can they predict the structure of the protein, and can they solve an interesting scientific problem? So one of the early tests that we did was the comparison of the players to a purely computational algorithm. And in this case, we compared the Foldit players to sort of the standard state-of-the-art algorithm when we ran this, which was called rebuild and refine, which is a standard Rosetta algorithm. And we looked at 10 protein structures. So each of the rows here is just a different protein structure. And we compared the RMSD, which is just a measure of how accurate the structure is, how far it is essentially from the right answer, from the what's called the native structure. And so lower numbers are better in this case. And so what we found was for this test set, the players were actually able to do better in 5 out of the 10 cases, similar in 3 out of the 10 cases, and actually worse in 2 out of the 10 cases. And so we took a closer look at what the players did and how they searched through sort of the space of structures differently from a purely automated method. And so in this chart here, every dot represents a protein structure, and the green dots are player solutions. The yellow dots are solutions produced by the rebuild and refine algorithm. And the chart sort of down is better energy or higher score. So the energy is the inverse of the score. So lower is a better energy or higher score, and to the left is a better quality structure, closer to the native, a lower RMSD. And so in this case, we can actually see the path that one player took to get from where they started here on this plot. Basically, they followed this blue line here through this space of structures, making the structure that they started with, which is this structure here. Actually, no, sorry, this is the starting structure here. They basically took this blue tail region, which was hydrophobic, and they unraveled the whole protein, getting a much worse score, and then they kind of refolded the whole protein with this blue, dark blue tail region in the interior of the protein, which is where it would prefer to be because it's hydrophobic. And so we called this move a strand swap because these arrow kind of regions are called strands, and the player essentially swapped the two different strands. And so this little kind of group here of structures was basically only found by players. And we believe that's because the players were willing to kind of unravel the entire structure and find these better energy structures basically by making the structure worse before it got better. And in this case, what they're doing is essentially escaping this local minima here in the energy landscape, where you can see there's kind of a notch here in the energy landscape where you have to actually get worse in the energy before you can get better. And so the automated algorithm solutions never really made it over that notch in the landscape, and they didn't really escape the local minima. And so that's why we think the players are able to find better structures, because they're willing to, they sort of have a plan, an idea about how they want to restructure the protein, and they're willing to have an energy that gets worse before it gets better. Whereas some of these automated methods, they will oftentimes give up if the energy gets much worse than what they've seen before. And so this was kind of a test set that we had given to the players to work on to see how they would compare. And so given that the players actually did well against the purely automated methods, we wanted to give them a real world scientific problem to solve. And so we actually were working with some collaborators who are trying to solve the structure of the Mason-Pfizer monkey virus retroviral protease, which is a protein that plays a key role in the virus that leads to AIDS in rhesus monkeys. They had actually been working on solving the crystal structure, essentially, of this, this protein experimentally for over a decade. And the computational methods that they had tried the state-of-the-art computational methods that they had been trying as well had also failed to solve the structure. And so we took this structure and we gave it to the players for 3 weeks to see how they did. We gave them this red starting structure. They already had a rough idea of what the structure looked like, but it wasn't as accurate as they wanted. So what happened was that in the 3 weeks that we gave it to the players, they were actually able to come up with a structure in the game that was close enough to the correct structure that it could be the experimental data that the scientists had could be used to sort of validate that the structure that the players had found was indeed the correct structure, and that they had essentially solved for the structure of this protein that was previously unsolved and unknown. And one of the most interesting things was that it wasn't actually just one player, but it was actually a team of players again. The Foldit players are able to work together, form teams, and build off each other's work. It was a team and 3 players working together in that particular case that were able to come up with a structure that was accurate enough to be able to say that the protein was solved. And so you can actually see, now that we know the right answer, and we can show it in blue, and you can sort of see how each player kind of brings the structure closer and closer as they work on it to the correct answer, what was later shown to be the native structure of the protein. So we thought this was really interesting that multiple players are working together, and multiple players working together are actually able to do some really interesting and amazing things. In the interest of time, I'm going to skip the algorithm development section and go right to protein design. And so in this case, protein design is in some cases the inverse in a way of structure prediction. Structure prediction is if you have a sequence of amino acids, what is the 3-dimensional structure of that protein? Protein design is kind of the inverse, where we say, well, if we have a particular function that we want a protein to take, and a structure that would, and can we figure out a structure that will do that, and what is the sequence of amino acids that will then build that structure, that will then fold up into that functional structure. And so to support protein design in the game, we have added a number of tools that essentially allow the players to change the amino acid composition of a particular protein. They can add and delete amino acids to the protein. And in particular, they can change the identities of existing amino acids. So there are tools like menus that come up when players can select a residue and actually change the identity and the kind of the atoms that make up a particular part of the protein to try to change the structure of it and improve the quality and the functionality of a particular protein within the game. So one of the problems that we gave to the players was actually to redesign an enzyme that carries out a reaction that's called the Diels-Alder reaction. And so in this case, there's this small molecule here that the protein enzyme is intended to catalyze a reaction for. And so we have the small molecule in here so the players can see it. And then the players are actually able to redesign this kind of brightly colored region on the side here in this particular puzzle. And this grayed out region on the other side, the players were not able to change. That was essentially locked. So the players had a little bit of freedom to redesign some regions of the protein. And what we asked the players to do was to redesign this region of the protein that they could change to try to make more contacts with the small molecule and hopefully make a better enzyme, a more efficient enzyme. And the really exciting part about doing protein design in Foldit is that we can actually experimentally validate the designs that the players come up with by synthesizing them in the lab. And so in this case, in this particular puzzle, we actually ran, or for this particular problem, we actually ran a series of several puzzles, about 4 or 5 different puzzles where we would post a problem to the players in the game. The results from that would come back. The biochemist we were collaborating with could then synthesize the designs that the players made, kind of bringing them into the real world, bringing them out of the game and into the real world, seeing how they work, making adjustments, working on them themselves a little bit, and then essentially when they felt that they could use some more feedback or some more ideas or some more input, they could then take what they had, post that as a puzzle to the Foldit players, and kind of repeat the process. And so again, we did that about 4 or 5 times on this particular puzzle. And in the end, we ended up with a player and scientist's co-designed enzyme that, starting from this scaffold, ended up with this blue structure here, where the players and the scientists essentially added these 2 kind of little helical regions here, which you can see make a lot more contacts or bring the protein structure a lot closer to the small molecule here that we asked them to design towards. And it turned out that this newly designed protein enzyme structure was about 17 times more efficient than the structure that we started with. And the players actually added about 13 new amino acids to the structure to make the protein actually quite a bit longer than it was originally. And the biochemistry we were working with said that it was such a large change to the starting structure that they themselves might not necessarily have considered it in the first place due to it being such a large change. But the players actually tried it out, and it seemed to work well in the game, and so it actually ended up working well in the real world when this enzyme was actually built and synthesized. So some of the next things that we're looking at with Foldit include support for multi-touch input. And this is a prototype of a project that we're calling Foldit Remote Control, where you can run the game on your desktop, so you get all the power, all the computing power that you have on your desktop to run kind of the optimizations and do the number crunching necessary to model the protein. But you can get the interface, the nice interface of a touchscreen or a tablet, and hopefully have kind of support for multi-touch kind of gestures, so you can actually pull on the protein in more places than one. You're not just limited by kind of mouse and keyboard, one pointer input. And so we hope that this will allow kind of new ways of interacting and reshaping the proteins. We've also got some preliminary work looking at the design of small molecules and drugs. As the previous example I was showing, where the players were able to redesign the protein to interact with the small molecule better, we're also starting to look at doing the inverse of that, where we can allow the players to actually redesign the small molecule to interact with the protein better and see what kinds of other interesting, effective molecules the players can come up with. And I'm really briefly going to go over some of the things that we've seen players producing in Nanocrafter as well. To go back to Nanocrafter a bit, this is one of the designs that the players came up with that is a multi-way junction. And so we asked the players to build 3 strands that all interact with each other once they are simulated. And so this is what a player came up with. And so this is kind of an interesting structural kind of subcomponent that could be used. We also asked the players to build a small set of strands that assembles into essentially a repeating structure when it's simulated. And so this is something that a player came up with. And this has basically a bunch of copies of 2 different sets of DNA that then self-assemble into this repeating polymer structure. And this one is more of a kind of an exploit of the physics system of the game, but this is something that a player built that kind of swims across the screen once it's simulated. So it's kind of a fun design that a player built. Probably not realistically plausible, but it's sort of a fun use of the physics layout system in the game that a player came up with. And so what we're interested in getting to the point where players are building kind of systems like this. This is an example test that we built. This isn't player built, but this is something that we built to kind of test out the system. This is actually a walker. So if you see there's this kind of arc here, if you can imagine these are 2 legs, and this is maybe like the body of a walker. These fuel pieces of DNA will come in and displace the legs, and the walker takes kind of a half of a step here. So this back leg gets displaced, and then it moves up to the front, and then the other leg gets displaced. And if there was more track, the walker would continue walking along. And so the players aren't at the point yet where we're seeing them build these kind of structures. That's what we're hoping that they get to. They have shown that they're building some basic kind of structural components, polymers, and those kinds of things. They're not yet, the players are not yet building things that scientists don't already know how to build, but it's interesting to see that they're figuring out how to build some of these subpieces and components. And so, of course, in the future, we're very interested in Nanocrafter supporting player creativity, doing some help automatically generating hints, potentially, for those tutorial levels based on how other players solve the levels, so that we don't have to manually author all of that content. Letting players share and customize these kinds of subcomponents that they can build so that they don't have to start from scratch every single time. And also kind of incentivizing players and training them to recognize and rate good solutions that they see other players producing. So in conclusion, I think we're just sort of getting started using games to combine humans and computers to be able to solve really challenging problems that neither humans nor computers could solve alone. And I think games are a really promising approach to trying to combine humans and computers together in a way that can engage and train players in solving difficult problems. Thanks for your time and attention. Both the games Foldit and Nanocrafter are freely available online. So if you'd like to give them a try, you can check them out at their websites. And of course, I want to thank as well the Foldit and Nanocrafter teams who put a lot of work into both of the games, and all the players as well. So that's it. Thank you. I don't know if we have time for questions, but I think I'm right at 10 o'clock.
Yeah, just a quick question, Seth. You mentioned for the multi-touch option you were going to add in, for the future. Had you considered the idea of maybe using leap motion? Because I saw you did.
Actually. Yeah, we actually have a Connect version and a Leap version as well. One of the difficulties that we've had with essentially both of those is that we found they're kind of a bit noisy and a bit imprecise.
Yeah.
So we've tried, if you do like a grasping kind of gesture and you're manipulating a protein, you sort of want, the cost of recognizing a grasp or a release, or not, having some noise in where the player's hand is relatively high, because it can it can have a big impact on the overall structure of the protein, if that makes sense, right? Like the mouse is nice because the mouse is very precise, and as to where it is and whether you've clicked or not, or released the button. And so if you're trying to make sort of small adjustments or even relatively large adjustments to the protein structure, the mouse is pretty good for doing those kind of repositionings. But the downside is there's only one mouse, and you might want to actually, if you want to bring two pieces together, if you're dragging one piece over with the mouse, the other piece might actually get kind of like repelled by that. So we actually have ways to get around that with rubber bands, but you have to set up kind of a system of these rubber bands before you can actually do those kinds of things. So if you could just directly kind of reach in and grab a couple pieces of the protein and move them together, that might be a much faster way to do that.
Yeah, great.
But it's a good question, and we have looked at Leap and Connect as well, but we've sort of run into technical difficulties with those in terms of kind of the precision and the accuracy that we could get.
Right. Any more questions?
Actually, with the whole feature.
Tyrone, can you come to the front so we can?
Yeah, sure.
We can get.
So yeah, you have the history, the future of the whole thing, and you have the DNA replication and the protein folding. Are you simulating the behavior of it as well? Does it, or is it just to replicate DNA that you could use in real world environments?
What do you mean by simulating the behavior?
Like, for example, with DNA, eventually some of these DNAs are used to change the behavior of certain cells and how they react within a nuclei. Could you predict that? Could you actually simulate that at all?
Yeah, potentially. Right now, the DNA simulation doesn't take into account sort of other potential molecules in the cell. The simulator that we're using, and so that's kind of an interesting challenge or problem, I think, is how would it behave actually in the in the environment, not just like in the lab necessarily environment, but sort of like in an organism or something like that. And again, just to clarify, I'm not sure if this is totally clear from the talk, but the DNA is not encoding any sort of like genetic information or anything like that. It's essentially just being used as kind of like a building block or something that a piece of material that can be modeled how it's going to interact. So
if you were to
the
make
actual sequences. Oh yeah.
Oh, so if you were to make an actual DNA strand, it's thousands and thousands, even millions and millions of lines long. So I can't expect people to make a whole version, but I was just wondering if it was possible to get ones that people made out there, bring them together and simulate kind of outcomes and behaviors of how certain cells would work if they were made.
Yeah, I mean, that's essentially what the game is. When you hit play, the game is simulating what the DNA would do if you actually built it in the lab, right? So the hope is that, if players build something that does something interesting in the game, for example, like that walker that I had demonstrated, that it would do something like that if it were synthesized in the lab. And then you could imagine synthesizing in different environments, something that, just like whether it's in a cell or just sort of like in a completely kind of like neutral environment as well. And so right now we haven't, the simulator in the game is sort of not considering necessarily what the interactions would be with other potential molecules, say inside an actual cell or inside an organism.
Yeah. Well, I only bring it up because, especially within the gaming world, let's just say if you ever played, I think it's a EA game where evolution of like cell into something. Sport.
Okay.
In sport, like a lot of people saw their creation come to life, and it's good to see that if they were heading towards that kind of direction, anybody would, a lot of that audience would probably just come straight over and get on board. Really, I just thought it's just like a wider market.
Mm-hmm. Yeah, I mean, we're definitely interested, as I was saying at the end, in letting players sort of make components that they built and share those, and build on top of each other's work. Yeah, maybe building larger and larger structures to the point where maybe you even abstract away some of the actual, not, I guess, kind of like Spore, right? Like you build the smaller the things, and then you sort of move up a level, and you're working at a kind of like a higher level of abstraction.
My last question for myself, actually, sorry if I'm taking up your time. You mentioned genetics. Do you think you'll ever go into RNA and help with transmission and messenger and all those types of, would you go that far, or is it strictly just protein folding and DNA makeups?
At this point, we're just looking at those. There actually is a game. There is a game built based around RNA called Eterna, which is also freely available to play online. So you can probably Google the name and find it. And that game is built all around essentially RNA folding and designing sequences of RNA that fold up into particular RNA structures, basically. So there are people who are looking into that and building games around that as well. Yeah.
Cool. Thank you. I think we have one more question. Just one moment.
Nowhere near a technical question, this, as they were just words. I didn't understand anything that he just said. I'm quite interested in the idea of gamification, and I noticed that one of your stats showed that 80% of your players were male. Do you have any ideas as to why it is that games seem to attract males more, and how we can attract more female players?
That's a good question. I'm not really sure I have a good answer for that as well. We tried to make a game that was, broadly appealing, and this is sort of the demographics of the people that came to play it. So yeah, I'm not sure that I really have a good answer to that. What do you think?
I don't know. I mean, I've tried employing games in class. I mostly teach IT and business type things, and it is usually the males that are more interested in playing the game, and they seem to sweep the females quite often to the side, and it tends to miss the point of the game, the educational aspects of the game, especially for the female players. And I'm at a loss really at how to solve that at the moment.
Yeah.
I was hoping you were going to have the answer.
No, I don't know. Yeah, I think if someone does have an answer, that would be great.
The question of the moment, isn't it?
It certainly is, yeah. I can't really employ it unless it brings all of the students together.
Mm-hmm.
Hey, right, I think we're running out of time, so we'll just, thank you, Alan. We'll just wrap up quickly.
Seth, we just wanted to say that we really appreciate your time today, and a big thank you from the Disruptive Media Learning Lab. I think Sylvester's got a little souvenir for you to say thank you from us.
Oh,
yeah, I've got something. Here you go.
Thank you.
I think it's a pleasure to have Seth anyway, because I've followed this work like since Foldit first started everything, and very impressed, and the fact that Seth is here now in Boston as well. It's just fantastic. It's like just at the right time.
Show us your face, Sylvester. Oh, there he is. Hello.
Well, yeah.
Well, thank you very much.
Yeah. Thanks,
We'll,
bye.
I'll leave it there. Thank you.
See you guys.
Bye.
Bye. Bye.